Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Ivan from Business Review, and I will be your host. It is our pleasure to have MedPace with us today, who will be presenting this webinar entitled Getting Ahead of the Expanding Landscape, Radiopharmaceutical Dosimetry. Today's guest speakers are Dr. Jess Guarnaschelli, Dr. Alexia Dost, and Dr. Michael Payne-McGlenn. Before we begin, I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin this session. In order to ask questions, you can send them in via the questions widget. Just type them in the box at the top right-hand corner of your screen, and then click Submit. We'll allocate some time at the end of this session to address any questions or thoughts that you may have. Please use the yellow Help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now, please allow me to welcome Dr. Guarnaschelli. Over to you.
Thank you, Ivan, and thank you for your time. It is a pleasure to have the opportunity to speak with you. My name is Jess Guarnaschelli. I'm a radiation oncologist and work in radiation clinical trials at MedPace. Like much of our work at MedPace, with advanced therapies, we focus on making complex trials seamless. Our MedPace Radiopharmaceutical Group views radioligand therapy as the most exciting and important example of these options for solid tumor patients. And today, my imaging colleagues, Drs. Alexia Dost and Michael Payne-McGlenn, and I will highlight key learning objectives. First, we will define dosimetry. We will touch upon old school classical radiation therapy techniques such as external beam radiation therapy and brachytherapy. We will review considerations for radiopharmaceutical therapy dosimetry, beta and alpha emitters, diagnostics. We will touch upon methods of dosimetry for novel radiopharmaceutical therapies such as absorbed fraction methods, S-value, MIRD, voxel level dosimetry, direct Monte Carlo methods, dose point kernel methods, voxel S-value method, and bioeffect modeling. We will discuss imaging challenges and site qualification, calibration challenges in getting sites up and running to perform radiopharmaceutical trials. I'd like to give you core knowledge to understand radiopharmaceutical dosimetry better. Radiotherapy is high energy particles, protons, neutrons, photons, electrons, etc. These are directed at cancer, and upon hitting the cancer cells, they are like tiny bullets. They cause single and double strand breaks of DNA and cause cancer cells to subsequently die or apoptose. Radiation therapy has been used for over 100 years. It is actually the oldest cancer therapy, and there are exciting recent innovations in how we administer it. The interesting thing is that the role of radiation therapy has actually increased with time in treating solid tumor patients. Because its radiobiologic effect and mechanism is predictable and known to differentially kill cancer cells, it is anticipated the application will continue to expand. Usually when you have a medical intervention that has been around for a long time, you see its use diminish as it's replaced with better, more efficient modalities. But this is not so with radiation. As technology and innovations have improved, we found more ways to use it. This also means we need improved ways to apply it safely on a global scale without excess toxicity to the patients we're trying to help. So it's in this context that technologies such as improved radiopharmaceutical dosimetry will make a positive impact. We're constantly trying to concentrate radiation dose to cancer cells while avoiding normal structures. And with these dosimetric goals in mind, we structure radiopharmaceutical therapy clinical trials that can be scaled to larger populations. Dosimetry is at the center of our big question. How do we confirm that our radiation is accurately being deposited where we intend it? Radiation absorbed dose and dosimetry serve as surrogates for anticipated efficacy and toxicity. The term dosimetry may refer only to the measurement in a radiation field, of a radioactive source, or of a radiation beam. It can refer to methodology for calculation of a dose or a dose rate value in a given medium, or it may also reflect the full chain from imaging, reconstruction, to dose delivery, and then also include the prescription dose. We focus on dosimetry in the process of radiopharmaceutical therapy. A dosimetry system consists of 3 parts. First, a set of rules to distribute the source inside a defined target to achieve a clinically acceptable dose distribution. Second, a method to calculate patients' tumor and organ at risk radiation dose. And third, a system for dose prescription. These steps were acknowledged and well-defined for external beam radiation and brachytherapy decades ago, in an era when computers were not available and sophisticated imaging tools like CAT scans, MRIs, and PET scans had not yet been integrated into radiation procedures. Extrapolating from external beam radiation and brachytherapy, implementation of modern resources with accurate calculation techniques and imaging procedures will allow us to further improve outcomes for radiopharmaceutical therapies. I'd like to take a moment to address the importance of units for measuring radiation dose. The quantities and units of radiation dose are inherently more complex than those used in toxicology or pharmacology, and additional complexity has resulted from changes required by evolving concepts in radiation dosimetry. System International units were adopted by the International Commission on Radiological Protection in the 1970s. SI units have been adopted internationally and in the US scientific community, but they were not embraced as enthusiastically by the US regulatory communities. Absorbed dose is the energy imparted by ionizing radiation per unit mass of irradiated material. The units of absorbed dose is the gray. The SI unit is the gray. The sievert is the SI unit of any quantities expressed as dose equivalent. The dose equivalent in sieverts is equal to the product of the absorbed dose in gray and the quality factor. So the gray and the unit of equivalent dose, effective dose, the sievert, each of those units equals 1 joule per kilogram. It's important for those without a radiation background to not get the sievert, which is effective dose, or gray, which is absorbed dose, confused with activity. Activity is the rate of disintegration or decay of radioactive material. The units of activity are the Curie and the Becquerel. Here is the weaponry board for radiation therapy. There is diagnostic cancer imaging, but that is not a focus of this webinar. There are many techniques for delivering radiation to solid tumors. These include external beam radiation therapy, brachytherapy, and of course, radiopharmaceutical therapy. At MedPace, we help our sponsors conduct trials with both radiation devices and radioligand radiopharmaceutical therapy and diagnostics. I will go through each radiation therapy delivery modality. Fortunately, radiation oncology has a long history of dosimetry-based treatment planning experience from conventional radiation tools with many techniques, personnel, and infrastructure readily adaptable to newer radiation modalities. For external beam radiotherapy, how do we validate that the aim from a linear accelerator is accurate and we are achieving the best possible therapeutic index, with radiation being concentrated into the tumor and avoidance of radiation to critical organs? There are a couple of ways one can predict and determine this. One can model an anatomic map of where the radiation is deposited in a patient if one knows from what angles and what intensities the linear accelerator beam is turned on. The lower right images show examples of these anatomic maps superimposed onto images. On the lower right-hand corner, these are axial images of a CT scan of the brain showing radiation delivery to the pituitary gland. The first are isodose lines showing radiation deposition from conventional fields with right and left lateral beam arrangements. The next to the right is from an intensity modulated radiation plan with 5 beam arrangements of varying intensity. And the one furthest on the right is from a VMAT, a volumetric modulated arc therapy plan, which delivers radiation from a linear accelerator using a cone beam that continuously rotates around the patient. These anatomic maps are useful for prediction, but does not tell you where the radiation was actually deposited. So the digital maps still don't tell us if the LINAC shot the beams where we predicted and intended them to go. For validation, we have phantom experiments. Phantoms are devices that can absorb radiation dose and show where the radiation ended up being deposited. There are many different types of phantoms. We can get outputs of phantom experiments which show that the radiation was where it was expected to be. These validation experiments are in physical space, whereas the anatomic radiation plans I just explained on the lower right of this slide with isodose lines are in the digital model. Then there is brachytherapy. Brachytherapy is a specific form of radiation used to treat cancer. It consists of placing sealed radioactive sources directly into or next to the tumor to be treated, either directly or by means of catheters, needles, beads, instruments, etc. Brachytherapy has been a part of radiation treatment since the early 1900s. A few years after the discovery of radioactivity, Pierre Curie and Alexander Graham Bell independently observed shrinkage of malignant tumors when radioactive sources were implanted directly inside of a cancerous mass. Throughout the mid-20th century, the use of brachytherapy increased and the technique became standard of care as a single modality or boost after external beam for tumors that required high radiation doses to be cured. Brachytherapy remains a tool in the context of radiation dosimetry. There are many applications for this, such as in cervical cancer, prostate, endometrial, breast, head and neck, skin, and many other solid tumor indications. There are many dosimetric advantages with brachytherapy as compared to external beam. There's a sharp radiation dose gradient, a sharp radiation dose fall-off compared with conventional external beam techniques. There's no beam going through the body, and thus the integral radiation dose is low. And as the radioisotope is near or right within the tumor, the radioactive source moves with the tumor, and thus there's no need for additional uncertainty margins around the clinical target volume. So conventional external beam radiation therapy requires additional margins for setup and organ motion uncertainties. Brachytherapy combines optimal tumor to normal tissue gradients while minimizing the integral dose to the rest of the body. The main limitation of interstitial and intercavitary brachytherapy is that it is invasive and requires short procedures either in the operating room or outpatient suites to plant the sources and the catheters or devices. Another key point of brachytherapy is that the quality of implantation is an important factor for success. Poor positioning of the sources may cause the tumor to be underdosed. Physicians and staff are more likely to receive unnecessary radiation exposure from brachytherapy as compared to traditional external beam. So given the logistical, scheduling, and radiation safety issues inherent to brachytherapy, there's an increasing temptation to propose alternative techniques that are easier to implement to broader patient populations with fewer logistical challenges. Most importantly, radiopharmaceutical therapy. In terms of radiopharmaceutical therapy, this consists of 4 key components. First, there has to be a unique target on the tumor cell. a ligand to bind it, a chelator or linker to attach the isotope, and finally the isotope or atom itself. Then there is the theragnostic paradigm, whereby administering a small pre-therapeutic activity of the therapeutic itself or a companion diagnostic agent with the same target, and imaging over multiple time points gives you more effective targeted therapy. In terms of mechanism of action, the radioligand therapy can be injected systemically and concentrated into target tumor sites, or can bind to receptors on the target cell membranes. Depending on the radioligand, it might remain on the cell surface or may be integrated and internalized into the cell. The radioligand then delivers radiation to the target cell, induces DNA strand break and cell death. The advantage of this is that it can be used for both imaging and/or therapeutic purposes using different isotopes on the same targeting ligand. And this can either be a diagnostic imaging isotope, a positron or gamma emitter, or a therapeutic radioisotope, such as lutetium-177 or actinium-225 or lead-212. So based on these principles, there are now a wide range of targeted radioligand therapies for oncology across the weaponry board. There are many dosimetric advantages of radiopharmaceuticals compared with new oncology drug entities. First is dosimetry. What you see is what you treat. You likely treat where you biodistribute. Imaging and early dosimetry may further expedite drug development programs by anticipating potential risks early in the game. Dosimetry can be used as a tool and a surrogate for toxicity and tumor response efficacy. Radiopharmaceutical dosimetry has suffered from early on from a perceived failure to demonstrate relevance and success in terms of predicting biological outcomes. And that may have been due to the fact that early on, there were imaging and dosimetry methods that were not quite as accurate, that they did not use complete methods. There were too few or poorly chosen time points. And previously, there were no calculations for uncertainties associated with absorbed dose in the literature. The European Association of Nuclear Medicine helped to rectify that problem when they published a methodology and guide to calculate uncertainties for dose calculations, and this greatly benefited the radiopharmaceutical therapy field. For tumor cell irradiation, there are some major differences between classical external beam radiation therapy and radiopharmaceutical therapy that must be pointed out. An external beam delivers the same absorbed dose per cell, regardless of the number of cells. But in radiopharmaceutical therapy, the absorbed dose delivered per cell by emissions originating from the cell is influenced by the range of the emissions, the number of cells that are clustered together, and the number of cells that have been targeted. If the range of the emitted particle is much longer than the dimension of the cell, a smaller fraction of the total emitted energy will be absorbed into the cell and into the nucleus. As noted earlier, current radionuclides in use and under investigation for therapy have varying emission ranges in tissue, and these are really important variables to consider. For example, the approximate emission range in tissue of lutetium-177 is 0.62 millimeters versus the emission range for actinium-225, which ranges between 0.05 and 0.08 millimeters. So this plays into the dosimetry methods. Please note that methods that are used depends on the study at hand. The S-value MIRD is the most well-established and commonly applied method. It is done no matter what for reporting and is useful as a QC guideline for the values of the other methods. Our dosimetrists do use the Monte Carlo methods combined and in parallel with voxel level dosimetry. Direct MC or Monte Carlo dosimetry methods are the most accurate and versatile approach for performing voxel level dose calculations. It's a statistical approach to solving the radiation transport equation. MC simulation models each physical process that affects the transport of radiation in a medium. Individual particles are tracked from the creation until the particle is absorbed. Direct MC simulations are more computationally demanding than other internal dosimetry approaches, and that's one thing to keep in mind. There are also the DPK, or dose point kernel method. DPKs describe the dose deposition from an isotropic point source as a function of distance from the source. They are obtained from MC simulations. DPKs were first limited to monoenergetic electron sources in water, but eventually isotope-specific DPKs were generated. Voxel S-value method was developed as an extension of the MIRD formalism from organ S-values to voxel level S-values. VSVs are calculated by integrating the DPK over the source and target voxels. The VSVs must be tabulated for each radionuclide, voxel size, source to target distance, and absorbing medium. That's a summary of some of the current dosimetry methodologies being used. Of note, ASTRO, AAPM, and NCI are all strong advocates for personalized dosimetry-based treatment planning, which is expected to become the new norm in the near future. And this will create a demand for qualified physicians, physicists, and clinical trial research teams to oversee and apply these methodologies and to know how to collect appropriate data for review. Preclinical testing for radiopharmaceutical therapy is important in that it identifies potential toxicities and is performed before the clinical trials. In radiopharmaceutical therapy development, normal tissue toxicity and dose-limiting organs are identified with preclinical testing in model systems. Compared with the given radiation activity, the absorbed dose to organs is closely related to toxicity. With these preclinical tests, the radiation activity is increased until toxicity is reached, and then the organ responsible for the dose-limiting toxicity can be identified through histopathology. Absorbed dose is calculated. All of this data is then used to design the phase 1 trials. Ideally, patient-specific 3D imaging-based dosimetry calculations should be integrated into phase 1 trials. Accurate dosimetry data reduces uncertainties in trials and eliminates concerns about potential toxicities and can help expedite the development process and entry into the clinic. As stated earlier, radiation oncology has a long history of dosimetry-based treatment planning tools and techniques and historic dose constraint values from older radiobiologic studies. Many of the current radiation dosimetric endpoints are extrapolated from historic radiation biology that dates back to the 1950s and 1960s. And these old dose constraints have not really been challenged. As things evolve and we design clinical trials, we must acknowledge the truth that patients enrolled onto these trials, many of them have already received previous therapeutic radiation from external beam or brachytherapy or radiopharmaceuticals, and thus the radiation cumulative dose should be acknowledged, it should be recorded, and it should be incorporated into the bigger picture of the patient's care plan. Currently, cumulative doses from external beam radiation therapy are not easily translated into the same dosimetric language for radiopharmaceutical therapy. Even with radiation-naive patients, it's important to set realistic normal organ dose constraints. Most often in clinical trials, the cumulative kidney dose should be less than 23 gray, and the cumulative bone marrow dose should be less than 2 gray. To date, we have not seen consistent dose constraints for brain and spinal cord for radiopharmaceutical therapies. We anticipate there will be an evolution in how we approach dose constraints with new beta emitters and alpha emitter therapies. Some takeaway messages. Radiopharmaceutical therapy is fast becoming a mainstream modality with the development and approval of new emitters and conjugates. Currently, FDA-approved radiopharmaceuticals are administered with fixed activities or using mass-based dosing. However, radiopharmaceutical therapies present an advantage over chemotherapy and other oncology new molecular entities with the ability to image the drug. Also, using the theranostic paradigm and imaging over multiple time points, the pharmacokinetic specifics to the individual patient are obtained, and the therapeutic administered activity may be adjusted to normal organ dosimetry tolerance limits, and this results in more effective therapy. Dosimetry is key and serves as a surrogate for efficacy and toxicity, and with the integration of image-based dosimetry, radiopharmaceutical therapy is likely to become the leading therapeutic weapon for treating solid tumors. With that, I will pass the slide on to Michael Payne-McGlenn for an imaging introduction.
Thank you, Jess. I'm Michael Payne-McGlenn. I am an imaging technologist with MedPace Core Labs, and for my background, I trained and practiced as a nuclear medicine technologist. So I'll be going over the imaging and the device qualification for radiopharmaceutical therapy trials. So nuclear medicine or molecular imaging are catch-all terms for the imaging of unsealed radioactive sources administered to a patient's body, which is the same principle that we use for radiopharmaceutical therapy. Different tracers can be selected based on their biochemical actions or on the physical properties of their isotopes. In this way, we can investigate different functions within the body, be that lung perfusion or the specific binding to a tumor-expressing receptor. Detection of the radioactivity is complicated and involves highly specialized devices. These depend on the type of radioactive emissions, and so we'll mostly be focusing in this presentation on the therapy isotopes and their emissions. So the radioisotopes that are used for therapy are imaged on a SPECT-CT or a gamma camera. Single gamma emission is random, and so therefore to make it unrandom or deterministic, the detectors have what we call a collimator. This rejects photons that don't come at the exact angle that is required to form a coherent image, and this design requirement of gamma cameras limits the resolution. And because it rejects 99.9% of the emitted photons, the sensitivity is very low. So it can take a long time to image a patient. That can be 15 minutes for a 2D whole body image and up to 45 minutes for a 3D SPECT-CT from skull to mid-thigh. Quantifying the activity with such low detection efficiency is a significant challenge. So SPECT isotopes emit photons across a broad range of energies. The different energy of the photons affects their detection. Higher energy photons can pass through thin collimators, and so the collimator must be thicker, which again further reduces the resolution. Some isotopes also, as you can see, have complicated decays and so emit more than one type of photon at high percentages. Lutetium-177 is an example that we will look at. So on the diagram, we have the detected response to lutetium-177 in a gamma camera. The low energy part of the graph on the left-hand side is the scatter and X-ray noise. The emitted photons make up the two major peaks. So while the photons emitted are always exactly the same, the response to it by the camera is less precise, and so an energy window is defined, and only the signal from that window is counted when composing an image. Quantitative imaging of the therapy isotope is the cornerstone of internal dosimetry. If we can't quantify how much, where, and when the tracer is, then there's no information to determine the amount of radiation that was delivered. SPECT imaging poses significant challenges because of its inherent limitations. And now I'm going to briefly pass over to Alexia, who will discuss how we adapt our practice at MCL to manage this.
Thank you, Michael. Hi, everyone. So I'm Alexia Daoust. I'm an imaging project manager. Very quickly, for my background, I have a PhD in the field of imaging. And today, during this webinar, I will talk over the operational aspect of dosimetry trial. So we'll start by site qualification for dosimetry and its challenges. So let's start by the feasibility questionnaire. So this document has to be sent to the recruiting sites at study startup, so usually very early on during study startup. So this document has to be done along with the clinical operation team, either by creating one together between the imaging team and the clinical operation team, or at least by reviewing the questionnaire of each other to make sure there is no repetition and no overlapping. This questionnaire is used to evaluate if the site has the appropriate machines and equipment, such as the appropriate collimators, dose calibrator, and gamma counter, but also used to know if the site has experience with the given isotope and if the nuclear medicine team at the site has experience with acquiring images the way we're asking them to do. And lastly, it is important to evaluate the level of English of the nuclear medicine department at site to know in advance if the imaging core labs need to translate the imaging manuals. So it happens that the PI at site speaks good English, but then the imaging technologists at nuclear medicine department do not necessarily speak good English. So it's important to evaluate this early on during the feasibility. In regards to site training and scanner calibration, following the completion of the feasibility and as soon as the site is ready, the MedPace Imaging Core Labs perform a teleconference training, ideally followed by a phantom scan done at site. And in regards to this phantom scan, in order to save time and money, we use the isotope of the mock shipment, if there is any mock shipment. We use this isotope to perform the scanner calibration, so to scan the phantom. Once this is done, if the phantom looks good, then the site can be qualified. From the end of the feasibility to site qualification, it usually takes a month for a trial involving dosimetry. So the scanner calibration now, this is done during the qualification, as I just said, it is important to understand that a dosimetry trial cannot start without scanner being calibrated. So the calibration of the scanner is used to assess the equipment setup and image quality, to assess the site capability to transfer images electronically to MedPace Imaging Core Lab, so to make sure they have installed correctly the database and they are allowed to upload images through the database, and most importantly, to calibrate the scanner for future dosimetry calculation. So the following qualification scans and data should be collected by the Imaging Core Labs, which include the gamma counter and dose calibrator calibration certificates, and the phantom scan using the stated isotope. And now I will pass it back to Michael for going deeper into the technique.
Thank you, Alexia. So as mentioned, the gamma counter and dose calibrator are two important pieces of equipment for quantifying radioactivity. The former for biological samples and at very low activity levels, and the latter in higher quantities like the prepared dose for administration. So the site's practice for quality assurance should be maintained and the records should be obtained throughout the trial. And for any new or unfamiliar isotopes, an accuracy measurement with a calibrated source may be valuable to verify the response of the devices to the isotope. Likewise, for the SPECT camera, performing a phantom scan with the phantom filled with the isotope to be studied allows the sensitivity and the response of the camera to be determined. This baseline measurement allows the quantification of activity in a patient. The activity within the phantom needs to be measured very precisely, and the phantom must be scanned on the exact same parameters as a patient scan. Following this, the parameters can be verified by MCL, but also the scanning protocol can be saved onto the camera ready for use with patients. So here is an example of the specific requirements for lutetium-177. So it would need a medium energy collimator, otherwise the high energy emission from lutetium would dominate the image. And also the two energy windows must be acquired and quantified separately because the two photons interact differently both inside the patient and inside the camera. If you only quantify one of the peaks, you're going to lose about half of the data, which would further reduce your detection sensitivity. Sites likely won't be familiar with this process of separating out the peaks, so it brings us full circle, highlighting the importance of both the training and the qualification scans to give them a dry run at this. So the specific needs of radiopharmaceutical trials, and in particular quantitative SPECT, requires a much more focused training and in-depth qualification activity. Once the site has practical experience of the scanning protocol, it can be saved to the scanner, and therefore the process for the patient imaging will be much smoother. And so on that note, we'll look into the patient's imaging. So when imaging patients for dosimetry, there are a few key differences compared to clinical practice. In order to determine the time that the isotope spends in the body, it needs to be imaged across a range of time points to produce the kind of graph that we have here. Early time points determine the peak uptake in the tissue, and late time points observe the washout. And each tissue, as you can see from the graph, has a different rate of washout. So having at least 2 time points in the middle gives a more accurate estimation. For therapy isotopes too, with longer half-lives, typically the imaging will be conducted over a week for a full set of dosimetry scans. The anatomical coverage can be flexibly focused on the extent of the disease if only evaluating the uptake in the tumours. However, one of the most exciting aspects of radiopharmaceutical therapy is that it's an effective treatment for highly diffuse metastatic disease. So in those cases, the tumour location wouldn't be necessarily predictable. In addition, for safety purposes, there are critical organs that will need to be captured. For example, the brain, heart, kidney, bone marrow. So a SPECT CT from skull to mid-thigh would give you maximal coverage, but it must be remembered also that 45 minutes each at up to 5 times is a lot of procedures for the patient, especially given the kind of patients that are included on early phase radiopharmaceutical therapy trials. Now, the specific imaging protocol for a dosimetry trial must be followed exactly. Unlike standard of care imaging, imaging on the wrong parameters will make a time point unquantifiable. And if the images are unsatisfactory, it's not possible to re-administer another dose of the therapy agent just to acquire further imaging. In training, sites are informed of the necessity to be quick with the data when turning it around, because if there are any safety review or dose adjustment committees to meet before the following treatment dose, the timelines for the dosimetry analysis are very short. So sites need to very closely engage with MedPace in order to anticipate potential pitfalls, and that can be done also during qualification. So one of the great benefits, as Dr. Garnishelli mentioned, is that using a single binding agent or ligand, you can attach that to either a diagnostic or a treatment isotope. So here in this example, we have PSMA, which is a prostate-specific membrane antigen, which has been a diagnostic vector with gallium-68 for a while, and that's the image on the left, but also it has been demonstrated as a successful therapy option when bound to lutetium-177, which we can see the anterior and posterior planar images on the right. So PSMA is overexpressed by prostate cancer cells even after they've spread throughout the body. And so we see normal uptake in the liver and the kidneys, salivary and lacrimal glands, but also diffuse prostate cancer cells in the spine, shoulders, pelvis, and multiple lymph node locations. Now, what we see on the gallium is where these cells are, and what we see on the lutetium is where exactly the therapy is being delivered. So a direct result of this principle is that we can use the uptake of the diagnostic tracer as a predictor for the uptake and therefore the dosimetry of the therapy agent. For quantifying the organ dosimetry, we need imaging at multiple time points, and then from the time activity curves, we can model to predict the absorbed dose from the therapy isotope based on the difference in the physical characteristics. The diagnostic isotope is, or tracer, is delivered at a much lower dose, and so is lower toxicity. So this is a very appealing aspect to the theranostic paradigm. Now, with any radiotherapy, the intention is to deliver the maximum amount of cell-killing radiation to tumors while limiting as much as possible the dose to healthy tissue. The activity must be observed over time from quantified images, and the absorbed dose will be derived from the time activity curve. In clinical trials, when investigating novel treatments in early phases, the intention is to gain evidence that the treatment is effective. So the highest dose that can be safely tolerated, the maximum tolerated dose, is likely to provide the greatest anti-tumor effect. However, with all radiotherapy treatment, the long-term aim is to optimize the dose to get the best treatment with the lowest possible dose administered, so as to minimize the long-term risks of radiation while still effectively controlling or eliminating the disease. And then finally, I hope in my section I've covered the key points that the inclusion of dosimetry into a clinical trial takes careful imaging considerations. Inability of sites to follow the imaging protocol can have a significant effect on the validity and strength of the data that's acquired. But ultimately, if we can find the dose of the product that keeps the patient safe while also treating them effectively, and using image-based dosimetry as a quantification tool, as powerful as it is, that will allow better and more dose optimization in the future. And so now I'm going to hand back to Alexia for her section on pharmacokinetics.
Thank you, Michael. So yeah, so we'll now focus a little bit more about pharmacokinetics and dosimetry measurements. So the pharmacokinetic is the study of the time course of drug absorption, distribution, metabolism, and excretion. And the main goals of clinical pharmacokinetics are to enhance efficacy and decrease toxicity of the investigational products. So simplifications of body processes are necessary to predict a drug's behavior in the body, and to do so, the pharmacokineticist applies mathematical principles to the various processes and must select a model. And there are two main different models. There is the compartment model and the non-compartmental model. So about the first one, the compartmental models, it includes the one compartment, two or multi-compartmental model. And then the non-compartmental model, also called NCA, this one is a gold standard that is mostly used for early phase trial. And it is used when data are being analyzed for the first time, usually. So pharmacokinetic analysis relies on observed drug concentration measurement over time within a biological sample, so usually blood, urine, and plasma. There are several steps for pharmacokinetic calculation. So it starts by, of course, the injection of the drug within the patient, and then biological sample collection. And from those collections, the biological samples are counted with a gamma counter. So the number of count per minute is being collected as per CPM, which is the count per minute, and then converted to a concentration in becquerel per milliliter. The pharmacokineticist receives this concentration and uses a software package that is Phoenix for the NCA analysis. And then the pharmacokineticist is able to construct the concentration versus time plots to graphically display the pharmacokinetic of the drug in the given matrix and to estimate the relevant pharmacokinetic parameters. So from these plots, the pharmacokineticist can begin to understand the absorption and elimination characteristics of the drug. So the typical pharmacokinetic parameters are listed here. So we'll start by the three main parameters that are usually collected for a radiopharma trial. It includes Cmax and Tmax, which are the peak concentration and the time at which that concentration is reached. As you can see in this graph, you can see the Cmax for the plasma concentration and the Tmax that corresponds to the time. And from this graph as well, you can see that we can collect the area under the curve, which is called AUC, and that represents the total drug exposure across time. So those three values are the most commonly asked for radiopharma trial. And then we also can collect the volume of distribution, VD, that is the theoretical volume necessary to contain the total amount of an administered drug at the same concentration that is observed in the evaluated matrix. We can also evaluate the systemic clearance, which is called CL, that is the measure of the drug removal from the body over time. And so here, the complexity of this calculation is for radiopharma study is that the compound decays over time, but in addition of being cleared from the body. So this is where the complexity happens for a radiopharma drug. And lastly, we can determine the terminal half-life, that is the time required to clear 1/2 of the drug from the plasma after reaching pseudo-equilibrium. So now a little focus on blood and plasma. So as we just discussed, the blood and plasma counts can be used for pharmacokinetic, but not only. It can also be used for dosimetry calculation. Let's start by an overview of the bone marrow, what it is. It is the most radiosensitive tissue in the body. All blood cells are produced in the red marrow and originate from one single type of stem cell. So the activity in the bone marrow, it can be determined from the activity concentration in the blood or plasma if there is no specific uptake in the bone marrow cells. And then more specifically into the blood collection. So the blood samples should be drawn from the arm opposite the injection site in order to eliminate any risk of contamination. And then this kind of the same kind of slide, but about urine samples. Again, it can be used for pharmacokinetic, but not only, also for bladder wall dosimetry calculation. So the bladder is the most exposed organ to radiation as it eliminates the radiopharmaceutical from the body. And the activity in the bladder wall can be determined from the activity concentration in the urine. So if the trial includes 3D images, such as SPECT or PET, then there is no need to collect urine sample for dosimetry calculation. However, if only 2D images are performed, then urine collection needs to be done at different time points, mostly before infusion, before imaging, and then after infusion for several days, depending on the isotope that is being used. So most commonly, a well counter is being used at sites. It's either a single or multiple wells gamma counter. And this is done at sites directly because the samples are radioactive. So the response of the detector of the gamma counter should be well characterized for the given radionuclide and measurement geometry, so that the corrections for sensitivity, dead time, and volume dependence can be applied. So that goes back to the importance of qualification of the site and making sure that the gamma counter is well calibrated. Also, the site has to use a fixed volume, meaning that each tube that is being counted should have the same final volume. It's what we call to have the same geometry. It also means that the site has to have a pipette in order to pipette the volumes. And lastly, the acquisition time has to be chosen so that the acquired background corrected number of count is higher than 100,000, so that the statistical inaccuracy of the measurement is less than 1%. So just to summarize what I've said about pharmacokinetic and blood and urine-based dosimetry. So the pharmacokinetic is the study of the time course of drug absorption, distribution, metabolism, and excretion. It uses biological samples, radioactivity counts done by the sites. The NCA analysis is usually used as a gold standard, and blood and plasma samples can be used for pharmacokinetic, but also for bone marrow dosimetry calculation, and urine samples, gamma count, can be used for pharmacokinetic, but also for bladder wall dosimetry calculation if the study only uses 2D images. And now we are moving to the last section of this webinar, which is about data management for dosimetry trial. So there are different data needed for dosimetry trial, but I will focus on the urgent data that we need for dosimetry calculation. So they are listed right here. Urgently, it is needed to have the injected activity value. Ideally, it has to be decay corrected, the date and time of scans, and the biological sample counts and time of counts, etc. So that's the gamma counts of the biological sample. So the site personnel entering those data into the main EDC should have a procedure in place and communication system in place to receive the data from the nuclear medicine department, because all of those data are collected directly from the nuclear medicine department, so they have to pass those data to the person entering the data into the EDC. It is also important that the CRA asks for source data as soon as possible, such as the injected activity. And it is important also to collect and verify the weight of the patients for the studies having pediatric population. So here is the list of risks and challenges of data collection for a dosimetry trial. So I will start by the first one, which is late image upload because of site standard procedure for image de-anonymization. So indeed, that can take few days or few weeks. So this is why it's very important to identify that very early on during the feasibility of the site. So we should ask what are their standard process, and if it's very long and the dosimetry data are needed urgently, then we can negotiate with the site to speed up the anonymization process. Secondly, the data entry delayed because of bad communication between the nuclear medicine department and the person entering data in EDC. So that goes back to my previous slide. And again, this communication has to be established early on during the trial. And again, during the feasibility or during the training, we can ask if they already have this communication in place, if they have standard procedure in place for communicating those data, and ask them how they do it, using which document, etc. And then late data entry for gamma counts. So from our experience, it is actually much better and much more efficient to collect those data within a spreadsheet rather than asking the site to enter the data directly in EDC. Because this procedure is done by the nuclear medicine department, and they are not the ones entering data into the EDC. So it's much easier to ask them to complete a dosimetry worksheet. We can also ask for source data, and then the dosimetry worksheet can be used and uploaded into the system to auto-populate the EDC. And to finish, the unverified data. So if we use unverified data for dosimetry calculation, that may lead to wrong dosimetry calculation and to wrong estimation of the injected dose for the next cycle or for the next patient. So again, this is something to be discussed very, very early on between the sponsor and the CRO or imaging CRO to understand the process and to see what is the best for this trial, and also take the risks into account. So this is a summary of what I just described. So this is the typical timeline for data collection for the dosimetry trial. So if we start at the top, we have data acquisition at sites, And then the site nuclear medicine department sends the images for anonymization. So that takes between 1 to 5 business days. It depends on the site. And then the nuclear medicine department transfers the data to the person entering the data into the EDC. And in parallel, they upload images into the imaging ECRF. So that can take between 1 to 10 business days. Then the CRA verifies the data. So that takes between 1 to 2 weeks. It depends on the CRO. It depends on the contract that we have established with the sponsor and the CRO. And then the dosimetrists can start using the data for dosimetry calculation. So as soon as they have all the data, verified or not, it takes about 10 business days to provide dosimetry calculation. So just to summarize, so the data used for dosimetry calculation have to be entered in EDC as soon as possible and be verified by the CRA urgently if the dosimetry data are expected within few days after the patient being injected, or be verified following the standard procedure timeline if dosimetry data are not required urgently. And also, it is important to take into account the possibility of data entry, image upload delay, and anticipate the risks at study startup. So this is the general conclusion of this webinar. So the following considerations are important for any radiopharmaceutical trial. The communication. So communication between the imaging core lab and the clinical operation team, and also the communication between the nuclear medicine department at site and the person entering data within the main EDC. Also, it's important, the scanner calibration and the site qualification is very important for this kind of trial. And the imaging protocol has to be strictly followed, as Michael explained you. And globally, as a very general conclusion, dosimetry and pharmacokinetic calculation are powerful tools and can help the physicians and their clinical decision for treatment dose. Thank you so much for listening, and I will hand over back to Ivan.
Thank you very much. Just a reminder for the audience, in order to ask questions, you can send them in via the questions widget. Just type them in the box on the top right-hand corner of your screen and then click submit. We already have received the first question, and it is, why choose SPECT CT imaging over planar imaging if SPECT takes so long and patient comfort is such an important consideration?
Thanks, Ivan. I'll take that. As Dr. Garnaschelli discussed during the dosimetry methods section, the most accurate methods that are available right now are the voxel-based and the Monte Carlo-based techniques. So these, unfortunately, can't be performed when you only have 2D imaging. It needs to be 3D images to be able to analyze the voxels. So, depending on the guidance of the FDA or whichever other board the protocol is submitted to, it may be possible to have a mixture of planar and SPECT-CT imaging, which will reduce not only cost but also the burden on the patients in the trial. That's a good question.
Okay, thank you for that answer. And we have our next question here, which is, can you please further expand on the dosimetry implications for alpha particle RPT? What are the specific challenges of alpha particle RPT dosimetry?
Sure, I'll take that question, Ivan. So one dosimetric challenge for alpha emitting radiopharmaceuticals is that the localization of activity uptake often leads to localization of absorbed dose at the sub-organ level. This complication is more prevalent in alpha emitters, where the short range of emissions implies that the absorbed dose follows the localization pattern of the activity more closely. Another challenge is that many alpha particle emitters have alpha-emitting daughters, which can relocalize after the decay of the parent isotope, and that requires separate activity localization measurements and separate dosimetry calculations. And then also the high potency of alpha emitters means that only small quantities of the radiopharmaceuticals are administered therapeutically. And it makes 3D imaging difficult because the low activity results in a low count rate. So all of these challenges for alpha emitters will need to be addressed to allow for a broad applicability in alpha emitter dosimetry. Thank you.
Thank you for that answer. And we have our next question here, which is, what is the difference between PK and dosimetry?
Yeah, I can take this one. This is Alexa speaking. So it is pretty much the same goal, but at different levels. So they are both used to define the treatment dose that is efficient enough, but also minimizes the toxicity due to excessive radiation. So the pharmacokinetic is used to measure the radioactivity in a specific matrix, as I said, like blood, urine. Whereas the difference with dosimetry is that it uses to measure radioactivity at an organ-specific level. That's the difference.
Okay, thank you very much for that answer. Unfortunately, we have run out of time, and we don't have any more time for more questions. So that just leaves me to thank Dr. Guarnaschelli, Dr. Daust, and Mr. Payne-McGlenn for what was a great presentation, and to MedPace for sponsoring this session. To the attendees, you'll receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. We look forward to sharing further webinars with you. So please do keep an eye out on the website just mentioned, and follow us on Twitter at BR Webinars for daily updates, and join our LinkedIn group Business Review Webinars. Thank you once again, and I hope that you all have a very nice day. Goodbye.
Thank you.
Thank you, everyone.